Hello and welcome everyone. In this podcast we're going to continue our discussion of the lac operon, but more specifically we're going to look at lac operon mutations. These mutations are really what led Jacob and Monod to be able to figure out the exact functions of the different parts of the lac operon. Everything we just talked about, the different genes, the different operators, promoters, inhibitors, and the ZYA genes, were all heavily mutated, and that is what allowed the scientists to figure out what those genes are. Let's just draw the operon here again. We'll get tired of drawing this. Alright, now remember there are several components to this operon. We mentioned before that there is this inhibitor protein, or inhibitor gene that makes this inhibitor protein. We then talked about the promoter, and then a new regulatory system that we hadn't spoken about before is called the operator. And the operator is always between the promoter and the gene, so that it can block transcription, block the RNA polymerase from moving forward. And then we had our three so-called structural genes. Z for beta galactosidase, then we had Y for permease, and then we had A for the transcytolase. So each of these could be mutated, and so let's just kind of go through this one by one. I'm going to erase this inhibitor here at the bottom. I can draw it at the top so we don't forget about it. If we had a mutation in Z, the beta galactosidase gene, so mutant here, so we'll call this Z minus, and then over here we'll write the phenotype. That is the expression. How does it affect the expression? So when the Z gene is mutated, the beta gal function for beta galactosidase, I'm just going to abbreviate it, no function. It doesn't work anymore. The permease mutation, Y minus, resulted in no permease. And this also meant no lactose could get inside of the cell. Before we move on though, let's look at our operon here. And so when we say Z or Y is mutated, what we're saying is that the operon can still be repressed by this inhibitor. It can still be pulled off, and you can still make the messenger RNA, but the proteins that are made will not be functional. Let's look at two other mutations now. The first one is I minus, and the next one is I with an S as a superscript. These both involve mutations in the inhibitor. The I minus mutant resulted in the genes ZYA always expressed, EXP for expressed. With or without lactose present, the inhibitor was always expressed. So let's talk about that one first. This result suggested that the inhibitor was no longer able to bind to the operator, because remember, when it bound to the operator here, it would block RNA polymerase from transcribing these genes, particularly in the absence of lactose. But now when you make this I minus mutation, even when no lactose is present, you're turning these genes on. So when no lactose is present, let's go ahead and remove this here, you would still make these gene products of Z, Y, and A. And so it's important that we think about this inhibitor protein. You probably heard this term before, but I'm going to say it again. Active sites. Proteins, enzymes, have active sites. This is where substrates bind. And if you mutate those particular active sites, it can no longer bind to the DNA. So, or any other substrate, depending on which active site you mutate. So in this case here, so let's say that this one here is the DNA binding, BIND for short, site. This is where the DNA binds to. This I minus mutant, because we're always expressing ZYA, tells us it's not bound there. We suspect then that this mutation has its DNA binding site mutated. So the I minus So this would be the I plus. Let's go and write that here. The way it would normally look unless it's mutated. The I minus now is going to look maybe something like this, where the DNA binding site has been mutated, so it can no longer bind to the operator. If it can't bind to the operator, you can't block transcription of ZYA. Now what about this other active site? Well, this is going to be our lactose site. The site where lactose binds. So when lactose is present in the cell, it could still bind to this repressor. It just doesn't do anything when it binds to it, because it's never going to be bound to the operator because the DNA binding site is mutated. Okay, let me erase this bottom one here, our example of the I minus repressor, and let's talk about this IS mutant. So this IS mutant, its effect is that it never, under any growth circumstances, expresses ZYA. And so we think about our inhibitor here that I've already drawn. Which site do you think might be mutated so that it it never allows these genes to be expressed? Turns out it's the lactose active site. And so when it's mutated, it changes its shape a little bit, like so, where the DNA binding site is still active, but the lactose binding site isn't active. So what does this mean? Well, that means that the inhibitor is still able to bind to the operator and block transcription of these genes. The difference is when you put lactose inside the cell, when lactose comes in the cell, because the lactose binding site is no longer functional, it can't bind there. And if it can't bind there, that means it can never change its shape to fall off to allow for transcription. So lactose enters the cell really wants to metabolize lactose, but it can't. It's a sad story, but it's true. It can't. So RNA polymerase does not have access to this operator because lactose can't bind to this mutated repressor. These genes will never be expressed. Next, we want to talk about some of these regulatory mutants. So let me erase everything down here on the bottom. Okay, we now have our operon back in place here, and let's talk about some other mutants here. And one of the ones they discovered that had a pretty strong effect, so again, let's write our mutants here, and then the phenotype. This one that they found that I want to talk about now was called an OC. O for operator, and C because it is constitutively active. Meaning that in this mutant, ZYA always expressed. So how will a mutation in the operator region allow these genes to always be expressed? Let's draw our inhibitor again. Remember, we drew it with a DNA binding site and then a lactose binding site. And so normally it's going to bind to this operator. What happens in these OC mutants is that the operator has been mutated and is no longer able to bind to the repressor. So let's just do this. Put an X through here. That operator is no longer able to accept that repressor. Now the repressor is perfectly fine. It could bind DNA if it had a place to go to. It just is no longer there. It's searching for it, but it can't find it. The repressor could also still bind lactose, but it's not going to have any effect because, again, this repressor only has a job if it can bind to the operator. If it can't bind to the operator, it's just floating around. So again, when we remove the operator in these OC mutants, we allow the continuous expression of ZYA. And the reason for that is because RNA polymerase now is unobstructed. It can bind to the promoter. It's never going to be blocked by the repressor and transcribe the gene ZYNA. Another mutation that they had was a promoter mutation. we just call this P minus. And this has sort of the opposite effect. In the P minus mutant, in fact, let's go ahead and erase some of these lines here. So in the promoter mutation, what we've done is mutated the promoter to the point where it is now no longer functional. It cannot bind to an RNA polymerase. So when the RNA polymerase is hanging around here, it's looking for that promoter, but it can't find it. So it can't bind to it. As a result of that, it doesn't matter if this repressor is bound to the operator or not bound to the operator. It's binding to it when there's no lactose in the cell, and it's falling off of the operator when there is lactose in the cell. But it doesn't really matter, because the repressor, even though it's doing its job coming on and off the promoter, it's not able to block the RNA polymerase because the RNA polymerase isn't able to do its job. It's not able to bind to here. And so as a result, ZYA are never expressed. Even though the inhibitor is doing its job by falling off the operator when lactose is present, the RNA polymerase is unable to do its job. Okay, let's go ahead and erase this. So now I want to ask the question of what does the cell do, or how are this operon expressed or not expressed when glucose is in the cell, or when glucose is in the cell with lactose. So I'm going to make this quick table here. So we have glucose, lactose, and then we have the ZYA genes of the operon. When glucose is present in the cell and there is no lactose, there is no expression of ZYA. That makes sense, right? Because without lactose in the cell, the repressor is going to be bound to the operator, so we would expect expression. And we also know if there's no glucose and we add lactose, that there is expression. Again, lactose comes in, binds to the repressor, comes off the operator, and we're able to express these genes. If neither are present, glucose nor lactose, then we get no expression. Again, for the same reasons. Because there's, as we saw up here, because there is no lactose. With no lactose, the repressor is going to remain bound. Now the interesting question is, when you have both present, do you express these genes? The answer is no. Your first thought might have been, my first thought would have been, sure, it's going to express these genes because lactose is present. Lactose is going to bind to the repressor. The repressor is going to fall off the operator. When that happens, RNA polymerase will transcribe ZYA and we'll have that operon expressed. But that's not what we see. That's not what Jacob and Minode saw. They saw no expression. So there must be a way to keep this operon turned off when glucose is present, independent upon the presence or absence of lactose. So let's spend a little time talking about this right now. Again, this observation isn't necessarily surprising in the fact that we know, or they knew at the time, that bacteria prefer to use glucose even if lactose is present. So it's not, in that sense, it's not surprising. But it was a little surprising once they worked out the mechanism of lactose metabolism, because it seems that it should be on because lactose is present. So let's erase this. And we're going to talk about a couple new proteins that we haven't mentioned before. This catabolite activating protein, CAP, and this signaling molecule called CAMP for cyclic AMP. And so we're just going to complicate the story a little bit, but it really makes for a beautiful picture here. So let's draw our operon, our world famous operon here. We have our Z, Y, and A genes here. We have our operator here, like we've always had, and we have our promoter here, like we've always had. So let's first talk about what happens when, or just remind ourselves what's happening when glucose is low. In this situation, let's go ahead and write it down. Up here, we have lactose at a high concentration, and glucose at a low concentration. Under this situation, just as we've seen before, the repressor binds the lactose, falls off the operator, RNA polymerase is able to make these genes. That's just half the story. And we might as well write it here as the first part. The repressor. Binds lactose to turn gene on. There's this second thing that must happen. There's an activator protein also present. And this activator protein also needs to be bind, so it binds near the promoter. And when it binds near the promoter, gene is on. And so let's draw this activating protein, CAP. When the CAP is bound here, it will activate transcription. So we need both things. We need the CAP, the activating protein, to recruit that RNA polymerase bound near the promoter. And we also need the repressor removed. And in a situation where lactose is high and glucose is low, this is what we see. Now I need to draw one other thing, because this cap protein can't bind to this area by itself. It can only bind to it when it has this signaling molecule attached to it, CAMP, cyclic AMP. This is the activating complex. The cap protein binds this small molecule CAMP. Even though I drew it as a rather large structure here, really the cyclic AMP is a very small component that binds to this cap. But it's an important one because it changes, it activates cap so that it can bind here. Okay, so just real quick again, when lactose is high and glucose is low, the repressor falls off. You can draw the repressor here. Falls off because lactose is bound to it. That's the first thing that happens. The next thing that happens is that the activator, the CAMP activator, binds here to recruit RNA polymerase to turn these genes on. Now let's change the story a little bit. What do we do, or what does the operon do when lactose is present and glucose is also present? Remember, the cell wants to use glucose because it's more efficient to use glucose. Let's erase what we have up here at top. So lactose is present. If lactose is present, that tells us our repressor is going to have lactose bound to it, and it's going to fall off the operator. Under the conditions we talked about early on the previous podcast, we would say this would allow ZYA to be expressed. But it can't be expressed because we also need our activator present. And it turns out that when glucose is present, our CAP protein is not able to bind to our cyclic AMP. And if the two are not coupled, if they're not bound to each other, it can't bind to the promoter. And if it can't bind to the promoter, it can't recruit the RNA polymerase. And if it doesn't recruit the RNA polymerase, it's not going to turn these genes on. So this is the way that the cell, the bacterial cell here, has come up with a plan, so to speak, to use glucose preferentially over lactose. This system pulls off lactose, binding lactose to the inhibitor, and once it's bound, it should allow these genes to be expressed, but it doesn't, because in addition to the repressor being removed, the activator here has to be bound to the promoter. And it can't bind to the promoter because CAMP and cyclic AMP is unable to bind to CAP. Now I should say, just to be in full disclosure, you might, in fact you probably would, just get a little bit of transcription from here. Not much, just a little bit. Very few genes are completely turned off. We almost always have a little bit of expression from many of our genes. Again, the reason it's doing this, I'm going to stress this last point, is it wants to use glucose first because it's a more efficient carbon source. Alright, now there's one more thing I want to talk about that's related to this, and that is, how does the presence of glucose keep cyclic AMP from binding to cap? Because that's what we're saying here. When glucose is present, cyclic AMP cannot bind to cap. If it can't bind to cap, it can't bind to the promoter. If it can't bind to the promoter, we can't turn these genes on. So how does glucose prevent cyclic AMP from binding to cap? I'm going to erase what's on the bottom here so I can draw this out. It turns out that we make cyclic AMP from ATP. And we do this using an enzyme called adenyl cyclase. So as long as you have adenyl cyclase in the cell, you're going to make cyclic AMP. As long as you're making cyclic AMP, it's going to be able to bind to cap. So what glucose does, by a mechanism we don't need to worry about right now, but as glucose is high in a cell, it inhibits this enzyme. Glucose is high in a cell, it inhibits this enzyme. We don't make cyclic AMP. If we don't make cyclic AMP, let's go ahead and erase that up here. It can't bind to cap because it's not there, and so cap is unable to bind to the promoter. So glucose is present, it inhibits adenyl cyclase. If adenyl cyclase is inhibited, we can't make CAMP, cyclic AMP. No cyclic AMP means cap can't bind to it. If cap can't bind to it, it can't bind to the promoter, and we can't turn these genes on. I recognize that this is a very complicated pathway, and there's a lot of players involved. As you're learning this pathway, I would focus on understanding the repressor aspect of it first. Make sure you have that down. When the repressor is bound, no transcription. When the repressor falls off, you have transcription. Understand that fully. What allows that to happen, what allows it to bind or not bind. Think about the mutations that we talked about with the inhibitor system. Once you have that down, then bring the activator into the picture. I think it's really hard to put this all together if you're trying to memorize it as one system. Learn each pathway separately. So then think about the activator. And think about how the activator is going to turn on or off transcription based upon the presence or absence of glucose. Draw this out. Have a friend quiz you. We'll do some activities in class to kind of help practice some of these problems. Because the more practice with this, the more understanding of the system, the more efficient you're going to get at it. So we'll definitely spend some time practicing this. Okay, I'm going to erase this and I'm going to close with a summary, but this particular summary I'm going to draw a table instead. I think it'll be a nice little practice for us. And so what I want to do here is draw this table and ask these questions. First one is going to be lac operon on. Is the lac operon on? Then the other question I'm going to ask is the repressor bound to the operator? And then the next question I'll ask, let's go ahead and number these, 1, 2, 3. So this last one I will ask is cap bound to the promoter? I say it's bound to the promoter, but it's really like near the promoter. But that's close enough. Okay, so I'm going to ask these three questions based upon these three conditions. Minus lactose, minus glucose. And then plus lactose, minus glucose. Plus lactose is our third condition, and plus glucose. If you can fill out this table, you understand the system pretty well. You can practice it on your own and go ahead and hit pause on the podcast and come back to it later and see if you got it right. But for now, I'm going to go ahead and go over the answer here. So if you're going to hit pause, go ahead and do it now. Alright, so for this first situation here, no lactose, no glucose. The lac operon will be off. The repressor will be bound to the operator, and the answer to that is yes. Is the cap bound to the promoter? The answer is yes. Okay. The reason that the repressor is bound to the operator is because lactose is absent. If it's absent, the repressor will be bound to the operator. So why is the cap bound to the promoter? The cap is bound to the promoter because glucose is low. Because remember, when glucose is high, we don't make cyclic AMP. But when it's low, we make a lot of cyclic AMP. When we make a lot of cyclic AMP, it's going to bind to the cap, and now the cap can bind to the promoter. Now, this next situation. Lactose is present, glucose is absent. Is the lac operon on in this case? And yes, it is on. Is the repressor bound to the operator? And the answer to that is no. It's not going to be bound because lactose is present. Lactose binds to the repressor and it pulls it off the operator, so the operator's not there. Is the activator, the cap, bound to the promoter? And the answer is yes, for the same reason as here. Low glucose, you're going to make a lot of cyclic AMP. If you have a lot of cyclic AMP, it's going to bind to cap. Cap bound to cyclic AMP will now be able to bind to the promoter to activate transcription. Okay, let's look at this last situation. When you have both lactose and glucose present, will the lac operon be on? No, it will be off. Is the repressor bound to the operator? The answer is no. It's not bound to the operator because lactose is present. It's going to bind to the repressor and pull it off of the operator. However, that's not enough to turn this gene on, because to turn the gene fully on, we need to have cap bound to it. And we can say that cap is not bound here. Cap, again, just to remind you, is not bound because glucose is high. When glucose is high, we block the formation of cyclic AMP. No cyclic AMP means it can't bind to cap. If cap can't bind to cyclic AMP, it can't bind to the DNA, to the promoter. So we can't turn on to expression of the lac operon fully. Again, go over this system several times. We'll go over some examples in class as well. If you have any questions at all, please make sure you come see me and I'll help in any way I can. Alright, I'll see you in class. Bye.